Picture this. We're in a lab, and a machine is listening to a voice.
Okay.
And the voice just speaks a single, isolated Japanese vowel, just like an ah or an a sound.
Right, just a very basic audio input.
Exactly. And the machine's job is to figure out which of 8 different male speakers is talking. And at first, it's basically just guessing. It gets it right about maybe 51% of the time.
Which is pretty standard. I mean, in the world of machine learning, that's a very familiar trajectory. You feed a system data, it processes it, it adjusts, and eventually it learns.
Right. But over the next 2 days, something wild happens. It gets better, like significantly better. It climbs to a 78% accuracy rate.
Wow.
But here is the twist. The quote-unquote processor running the speech recognition program, it's not made of silicon.
Right.
It's not a microchip at all. It is a 3-dimensional cluster of living human brain cells.
A brain organoid.
Yes, a brain organoid. It is literally sitting on this high-density multi-electrode array, absorbing electrical zaps, physically rewiring itself, and actually learning from them.
It's incredible.
It is. Welcome to today's deep dive. We have a huge stack of fascinating research in front of us today, and our mission is to basically explore this wild new frontier. We are asking what happens when our computers are no longer built, but grown.
Yeah, and it's an escalating journey that we're looking at today. It starts at the molecular level with DNA data storage, and then it scales up to these programmable bacterial logic gates, and it culminates in artificial intelligence systems powered by actual human brain tissue.
Which is just mind-blowing. But to understand why scientists are going to such extreme lengths to compute with biological materials, I guess we first have to look at the severe physical limits of the technology sitting on your desk right now.
Right, because for decades, we've really relied on Moore's law.
Yeah.
This whole idea that computing power basically doubles every couple of years as we pack more and more transistors onto silicon chips.
But I imagine we are simply hitting a physical wall at this point. I mean, You can only slice a silicon atom so thin before the laws of physics just step in and say no.
Exactly, yeah. We are absolutely scraping the bottom of the atomic barrel here. But even more pressing than the physical size of these components is the massive, almost unsustainable energy drain of modern artificial intelligence.
Oh, I bet.
The sources we have outline this perfectly. Training and driving a large artificial neural network on current AI hardware, it consumes about 8 million watts of power.
Wait, 8 million watts?
Yes.
Just to put that into perspective, that is the energy output of a small commercial power plant.
It is.
All of that just to run a complex AI model.
Right. And meanwhile, the human brain inside your head is doing vastly more complex, more dynamic and adaptive tasks on just 20 watts.
20 watts, that is like a dim refrigerator light bulb.
Yeah.
So why are our silicon computers so incredibly inefficient compared to a biological brain? I know data centers generate a ton of heat, which requires massive cooling systems. But where is all that base inefficiency actually coming from?
Well, a major culprit is a structural flaw that's inherent to almost all modern devices. It's known as the von Neumann bottleneck.
Okay, let's unpack this, because whenever I hear von Neumann bottleneck, I always picture a badly designed commercial kitchen.
Oh, that's a good way to look at it.
Yeah, imagine you have this massive pantry where all your ingredients are stored, and that's your computer's memory.
Okay.
And then way down the hall, in a completely different room, you have your chopping board and your stove, which is your computer's processor.
Right, and it perfectly illustrates the flaw, because to run a calculation, your computer has to constantly ferry data back and forth down that hallway.
Right, the chef is just sprinting back and forth, grabbing one ingredient at a time. And it doesn't matter if you have the fastest stove in the world, the whole process is completely bottlenecked by the time and the physical energy it takes to run down that hallway. Moving data generates heat, heat requires cooling, and all of that just wastes millions of watts.
And this is where biology wins, because biological neural networks entirely bypass that hallway.
Oh, really?
Yeah, in a human brain, there is no separation between the pantry and the stove.
Interesting.
The synapses between neurons, they act as both the memory storage and the processing mechanism simultaneously. The data is processed exactly where it is stored.
Oh, wow. So scientists are looking at biology and thinking, let's stop trying to force silicon to act like a brain and just use the actual materials of life.
Exactly.
But I assume you don't just start by hooking a clump of human brain cells up to a monitor. You have to start at the absolute foundation of biology, right?
You do. You start at the molecular level. So we have a paper in our stack titled A Primordial DNA Store and Compute Engine. which really lays the groundwork here. Because before you can process information, you have to actually store it.
Right.
And nature already invented the ultimate hard drive, which is DNA.
It makes sense. Instead of storing data as ones and zeros on magnetic tape, you encode it into the 4 molecular building blocks of DNA.
Right. Adenine, cytosine, thymine, and guanine.
A, C, T, and G become your binary code, effectively.
Yeah. And it is incredibly dense. I mean, you could theoretically store all the data on the entire internet in a shoebox of DNA.
That is just terrifying and amazing.
Right. And using specific enzymes, scientists can perform really basic computations, like strand displacement, where DNA molecules swap parts to perform simple logic operations.
But DNA is mostly just a passive storage molecule, isn't it? Like it just sits there waiting to be read.
Yeah, mostly.
So if we want a system that actively senses its environment and performs complex logic in real time, we have to graduate from isolated molecules to living cells.
Exactly.
Which brings us to a really cool 2024 Nature Communications paper by Fedorek and colleagues detailing an incredible leap in bacterial computing.
Yeah, and engineering biological systems to compute isn't entirely new. For years, scientists have been splicing programmable logic into the gene regulatory networks of single cells.
But I imagine the issue with just dropping a complex genetic circuit into a single cell is that you're going to completely overload it.
Oh, absolutely.
The cell is just trying to metabolize food and keep itself alive. It doesn't have the spare energy to run complex math for us.
Right. It causes a phenomenon known as metabolic burden.
Metabolic burden, okay.
Yeah, the cell becomes sick and sluggish. So to solve this, researchers tried dividing the computational labor. They created computational communities of microbes in liquid culture, basically breaking the complex logic into smaller modules across different bacterial populations floating together in a nutrient broth.
Divide and conquer. But wait, if they are all floating in the same liquid soup, how do they talk to each other without the signals getting completely crossed?
Well, that is the infamous wiring problem. To get these populations to communicate in a liquid, you basically need a unique chemical molecule for every single, quote-unquote, wire of communication.
That sounds complicated.
It is. Otherwise, the chemical signals crosstalk, the liquid becomes this noisy mess of overlapping signals, and the logic completely falls apart.
So a single cell is too weak, and a liquid culture is too chaotic.
Exact.
The solution they came up with is just brilliant, though. They took the bacteria out of the liquid entirely and moved them into distinct fixed spatial arrangements on solid agar plates.
Right.
They shifted from genetic computing to spatial computing.
Yeah, and by doing this, they lean into the physics of diffusion. Instead of trying to invent dozens of new genetic wires, they just let the physical space between the colonies do the heavy lifting. The information processing is basically encoded within interacting chemical gradients.
Okay, let's visualize this for everyone. Imagine a flat petri dish. The inputs for this bacterial computer are literal droplets of a chemical, a signal called IPTG.
Yeah.
When you drop this chemical onto the solid plate, it doesn't just sit there, it diffuses outward, creating a chemical gradient. So it's highly concentrated in the center where the drop hit, and it fades out as it spreads, kind of like ripples in a pond.
Right. And the hardware of this computer consists of engineered bacterial receiver colonies. So these receiver cells are programmed to respond to very specific concentrations of that diffusing chemical.
And this is where they use these really specific activation functions, right?
Yeah.
The paper talks about the bandpass biological mechanism involving a T7 promoter and a VHLF transcription factor. But let's translate that.
Please do.
It functions almost exactly like a biological brake pedal, doesn't it?
That's a great way to put it, yeah. At low chemical levels, the bacteria are completely dormant. But when the chemical concentration rises to a medium level, the engine starts, and the bacterial colony literally fluoresces green.
It turns on.
It turns on.
But if the chemical concentration gets too high, it triggers a secondary genetic switch, the brake pedal, that slams down and shuts the glow off entirely.
Exactly.
So only turns on in a very specific band of chemical concentration. Too low, nothing. Too high, it shuts off.
Right. And they also engineered high-pass receivers that only turn on above a certain threshold and low-pass receivers that only work below a threshold.
Okay.
The real beauty of this system is how physical position dictates the logic.
Think of it like an invisible chemical Venn diagram on the plate.
Okay.
You put down 2 droplets of chemicals, input A and input B. The chemical rings diffuse outward and overlap in the middle. The concentration in that overlapping middle section is additive, meaning it's the sum of both droplets.
Right, so if you place a high-pass receiver colony right in the center of that overlap, the concentration from just one droplet isn't enough to turn it on.
Oh, I see.
It requires the combined additive concentration of both input A and input B to actually light up.
Meaning, just by sitting in that specific spot, that bacterial colony has become a physical AND.
Yes.
It requires A and B to compute. If you move the colony a few millimeters to the left or change it to a bandpass receiver, it becomes an OR gate or an XR gate.
The position itself is the programming.
It's wild.
It is, and it elegantly bypasses the need to constantly re-engineer the genetics of the cell.
But if I want my bacteria to perform a really complex math problem, how do I know where to put them? I mean, I can't just guess where to drop them on the plate and hope the gradients overlap perfectly.
Right, so to solve this, the researchers developed an algorithm called macchiato.
Macchiato, like the coffee.
Yeah, exactly.
Yeah.
In traditional electronic chip design, there is a famous logic minimization tool called the espresso algorithm.
Oh, I see what they did there.
Yeah, espresso simplifies digital circuits into their most efficient layouts.
Yeah.
So macchiato is the biological equivalent of that.
Espresso for silicon, macchiato for biology.
You got it. It calculates the exact minimal set of bacterial receivers and their precise spatial mapping needed to build complex logic gates. You literally feed it a truth table, and it tells a liquid-handling robot exactly where to print the bacterial colonies and the chemical inputs on the plate.
That is so cool. And they use this to successfully build advanced spatial logic gates, including something called Rule 110.
Yes, Rule 110.
For anyone not deep into computer science, Rule 110 is a cellular automata rule set that is Turing complete. Meaning, theoretically, this arrangement of glowing green bacteria can compute literally any calculation that a modern silicon computer can.
Assuming you give it an infinitely large Petri dish and, well, enough time.
Right, time being the operative word there. But before we get to speed, we should look at how they applied this to the real world. Because robotically dropping chemicals onto a plate is a fun lab trick. But to make a standalone biocomputer, they upgraded the inputs.
They did. Instead of a robot dropping chemicals, they engineered biological sender colonies.
So these act as biological biosensors. But how do the senders communicate with the receivers down the plate?
Well, they use a mechanism called quorum sensing.
Oh, I've heard of that.
Yeah, in nature, bacteria use this to effectively count how many of their neighbors are nearby before they take action.
Right?
So the sender bacteria are programmed to release a quorum-sensing molecule, a specific one, only when they detect specific environmental inputs in the wild, like arabinose or lactate.
So the sender colony detects a specific chemical in the environment, say like a pollutant in a water sample. It shouts out a chemical signal. That signal diffuses down the plate. And the receiver colonies hear that signal and perform a logical computation based on where they are sitting.
Which points straight toward the development of diagnostic biocomputers.
Right, you could print a complex bacterial logic circuit onto a piece of paper. It sits dormant until you expose it to a biological sample, maybe a blood test. The senders detect the specific disease markers, the chemical gradients interact, the receivers compute the logic, and a specific spot on the paper glows green to say yes, this specific combination of markers is present.
It is a really profound demonstration of encoding information processing into physical space.
It really is.
But as we move through the history of computing, we always hit a familiar barrier.
Speed.
Speed.
Because as amazing as those paper-based bacterial logic gates are, they are so slow. The bacteria have to grow, the chemicals have to physically ooze across the agar, the genetic brake pedals have to engage.
Yeah, we are talking hours to get a simple result.
Right. That's incredible for a biosensor, but if we want to run AI, if we want to process human speech or navigate a self-driving car, we can't wait 14 hours for bacteria to diffuse.
No, definitely not.
We need a biological system that computes in milliseconds, which means we have to graduate from bacteria to brains.
Which brings us to a landmark 2023 Nature Electronics paper by Kai and colleagues, and the introduction of a hybrid system they call BrainAware.
Let's paint the visual for the listener here because it's straight out of a sci-fi novel.
It really is.
They took a human cortical organoid. This is a tiny pink spherical cluster of living human brain cells. It's a fully 3D structure containing mature neurons, early stage neurons, and astrocytes. It even develops early brain-like networks.
Yeah.
And they mounted this living sphere directly onto a digital chip.
A high-density multi-electrode array, or MEA. This chip basically allows them to send electrical voltage pulses up into the base of the organoid and record the evoked electrical neural activity coming out.
Wait, you lost me here a little bit. In a normal silicon chip, data follows a very specific rigid pathway, right? Like A connects to B.
Right.
With the bacteria, we literally place them in specific spots to control the flow. But a brain organoid is just a messy, tangled sphere of billions of connections.
Yeah.
How do you map a clean data pathway through that?
Well, you don't.
You don't.
Now, that is the genius of it. It relies on a framework called reservoir computing.
Reservoir computing, okay.
Yeah, instead of strictly defining the path, you map time-dependent inputs into a complex, non-linear physical system, which is the reservoir, and then you read the chaotic, high-dimensional output that system produces.
So it's kind of like dropping 3 different rocks into a chaotic pond and trying to figure out which rocks they were just by looking at the ripples crashing against the shore.
That is an excellent analogy, yes. The organoid acts as an adaptive, living reservoir. You take time-dependent data, like the audio clips of the Japanese vowels from our cold open, and you convert it into a sequence of electrical pulses.
Okay.
You fire those pulses into the biological neural network.
And because these cells are alive, they are chaotic. The paper notes they have fading memory, which I assume means a neuron is still ringing or echoing from an electrical zap it received a millisecond ago.
Right.
And that lingering echo physically changes how it reacts to the next zap.
Exactly. The network processes the overlapping data through those lingering echoes, and the MEA chip reads the ripples coming out the other side.
Whoa.
You then use a standard linear regression algorithm on a traditional silicon computer to decode those ripples and find your answer.
Which brings us back to the speech recognition test. They took 240 audio clips of 8 different men saying a Japanese vowel.
Right.
They encoded that audio into electrical zaps and fired it into the BrainAware. At first, it couldn't tell the ripples apart. It was guessing at 51% accuracy.
Yeah.
But over 2 days, it jumped to 78%.
Yeah.
It didn't just compute the data, it learned.
And that is the pivotal distinction between brain-aware and a fixed physical reservoir. I mean, a bucket of water doesn't change its structure over time. Brain-aware does.
Right.
They also tasked it with predicting the Hinnon map.
Which is a chaotic mathematical equation, right?
Yep.
Like trying to predict the weather based on a sequence of erratic data points.
Yeah, it is notoriously difficult to model. So they encoded the sequence data into pulses, and over 4 training epochs, the organoid actually learned to predict the chaotic sequence. Yes, its regression score improved from 0.356 before training up to 0.812 after training.
But how is a disembodied cluster of cells learning without a software update or, a programmer tweaking the code?
It relies on unsupervised learning driven by neuroplasticity.
Neuroplasticity?
Yeah, it's the fundamental property of the biological brain. The electrical stimulation physically reshapes the functional connectivity of the organoid's network. The neurons are forming new synapses, they're strengthening others, and physically reorganizing their communication pathways in response to the electrical inputs.
So the hardware is actually rewiring itself on the fly. As it computes, its physical shape changes.
Exactly. And the researchers proved this wasn't just some statistical anomaly with a really brilliant control experiment. They added a drug called K252A to the organoid.
K252A. So if I'm a skeptic, I'm thinking maybe this is just a fluke. Did adding the drug shut the neuroplasticity off entirely?
It did. K252A specifically blocks activity-dependent synaptic plasticity. So it basically strips the neurons of their ability to reshape their connections in response to stimuli.
And what happened to the learning?
The unsupervised learning completely flatlined.
Wow.
Yeah, the organoid maintained its baseline ability to compute the ripples, so it was still working, but it couldn't improve. It stayed completely stuck at its initial accuracy rate.
That's crazy.
Right, and this definitively proved that the biological reshaping, the physical neuroplasticity, was the exact mechanism driving the learning.
Okay, I have to stop here and ground this a bit because I know exactly where people's minds go when they hear learning human brain cells on a chip.
Yeah. Huh?
Is this a miniature conscious brain? Are these organoids thinking in that incubator? Are we biohacking our way to some sci-fi dystopia?
Well, it's crucial to dispel that hype immediately.
Please do.
This is not general AI,
Right.
and it is absolutely not a conscious entity.
Okay, good.
BrainAware is fundamentally a hybrid system. It is entirely dependent on traditional computers.
Oh, I see.
Yeah, a silicon computer is strictly required to encode the audio data into electrical pulses. and another silicon computer running statistical algorithms is required to decode the neural output. The organoid is just acting as a specialized processor right in the middle.
Right. And practically speaking, the engineering hurdles to make this a commercial reality are massive right now. You can't just buy a brain-aware chip at the electronics store.
Oh, no, not at all. The scaling difficulties are immense. For one, organoids suffer from high biological heterogeneity.
Meaning they're all different.
Exactly. If you grow 10 organoids, you get 10 slightly different networks.
Yeah.
So standardizing them for reliable mass manufacturing is a long, long way off.
Yeah, that makes sense.
They also experience necrosis and hypoxia.
Because they don't have blood vessels to pump oxygen to the center of the sphere, right? So the cells on the inside suffocate and die off, which is definitely not ideal for a piece of hardware you want sitting on your desk for 5 years.
Yeah, exactly. Plus, the MEA chip they're using is entirely flat. It only connects to the surface neurons of the 3D organoid, which means they are missing a vast amount of the computational power happening deep inside the tissue.
And there's an incredible irony here regarding the power consumption, too. I mean, we started this deep dive by talking about how the brain only uses 20 watts compared to an AI server farm using millions.
Right.
But to keep this tiny organoid alive, you have to run it inside a giant, power-hungry, humidified CO2 incubator. So we haven't exactly solved the energy crisis of AI just yet.
Well, no, not today. But the researchers do note that with future system integration and customized life support microfluidics, the overall power consumption could drop drastically.
That's fair.
What matters right now is the foundational proof of concept. They have undeniably established that living neural tissue can act as an adaptive learning processor.
It is a wild ride. Let's recap the journey we just took today. We started by looking at the energy drain of modern AI and the von Neumann bottleneck. We saw how scientists are using DNA as the ultimate primordial hard drive. We solved the bacterial wiring problem by encoding digital logic into the physical chemical gradients of E. coli colonies on agar plates, mapping it all out with the Macchiato algorithm.
Yeah, and then to overcome the speed limits of bacteria, we scaled up to brain-aware. We proved that human brain organoids can be integrated with digital chips to perform unsupervised learning, recognizing speech, and predicting chaotic equations through the sheer power of neuroplasticity.
In traditional computing, the hardware is a fixed, dead thing. Your silicon processor is the exact same physical shape the day you buy it as the day you recycle it. Only the software changes.
Right.
But in biological computing, the hardware is the software. As it processes information, it literally grows, adapts, and changes its physical shape to better understand the data it's receiving.
The structure of the computer itself is literally shaped by the data it experiences.
Which leaves us with a final thought to mull over. We are watching the barrier between biological life and machine logic fully dissolve. When that barrier is completely gone, what will the computers of our future look like? Will they sit silently on our desks, or will we have to feed them?
That's quite a thought.
It is. Thank you for joining us on this deep dive. If you enjoyed exploring this frontier with us, please like the video and subscribe to the channel. We've got plenty more mysteries to unpack. See you next time.